Yes, so what do we have today in our list? Someone say enzymes. Yes, I have scarcely talked about this enzyme, but I'm going to talk about it again, but in another form. What are these enzymes? Someone will ask. Enzyme can be defined as a biological catalyst. Then when we ask what is a catalyst? A catalyst is just a substance which speed up a chemical reaction but remain unchanged at its end. So when I say enzymes are biological catalysts, it's because they are protein molecules made by living cells. A typical human cell contains several thousand enzymes. They are used to catalyze a vast number of chemical reactions at temperatures suitable for living organisms. That's exactly between 5 degrees and 40 degrees centigrade. You see, high temperature will be needed as well as marked changes in other conditions. If the same speed of reaction were to be achieved outside the organisms, do you understand? That's when high temperature should be used. The chemical which an enzyme works on is called its substrate. So the chemical the enzyme works on, it is called its substrate. So an enzyme combines with its substrate to form a short-lived enzyme substrate complex. Once they combine, the proximity of these enzymes with the substrate in a complex greatly increases the chances of a reaction occurring. So once a reaction has occurred, the complex breaks up into products and enzyme. And the enzyme remain unchanged, like I told you, at the end of the reaction, and is free to interact again with more substrates. That's when enzyme is added to a substrate, it will give you enzyme substrate complex. An enzyme complex can still be broken down to enzyme product complex, and it can still be broken down to what? Enzyme plus products. I don't know how many of you understand this. So you can now say E plus X, which is enzyme plus substrate, is equals to, that's in, they are just reversible. They are reversible reaction. That's why when I say something is reversible reaction, the arrow will be here and there. So when E plus X is equals to EX, that's enzyme plus substrate is equals to enzyme substrate, and it equals to enzyme products complex, and is equals to enzyme plus products. Do you understand it? I wish you understand, but I will have ask room for question. I will have room for question. So before we talk about enzyme, we also need to understand what anabolism and catabolism is all about. I did not define when I mentioned anabolism and catabolism. So when you say the sum total of all the chemical reaction going on in cells, all the sum total, all of them, all the chemical reactions going up in the cell, is known as metabolism. And metabolism can be divided into two types. You can name it anabolism and catabolism. These two types of activity often take place in different parts of the cell. When you talk about catabolic reactions, it involves the breakdown of molecules and usually release of energy. And they also involve oxidation or hydrolysis. But then when you talk about anabolic reactions, it involves the synthesis of molecules and usually require energy. They often involve condensation. Now, catabolic, which means breaking down, involve what? Oxidation and hydrolysis. And anabolic reaction, which involve the synthesis, or you say making up, usually require energy, and it involve condensation. So all these reactions are catalyzed by enzyme. Now, Let's call example an enzyme that involve in anabolism. One of the enzymes that involve in anabolism is known as what? Glutamine synthesis. This glutamine synthesis catalyzes the synthesis of amino acid glutamine. Do you understand? When it's oxidized, when glutamine is oxidized, that's when it's being broken down. It will be broken down to glutamic acid and ammonia. Do you understand it? It requires energy, and the energy it always used is ATP, adenosine triphosphate. Do you understand? Then let's look at metabolic pathways. I told people, we have so many metabolic pathways taking place in the body. Many. The metabolic pathways, commonly a number of enzymes are used in sequence to convert one substance into one or several products through a series of intermediate compounds. The chain of reaction is referred to as metabolic pathways. The chain of reactions used in sequence to convert one substance into another. Not even only another, it can also involve into several products via a series of intermediate compounds. It's a metabolic pathway. So many such pathways are going on at the same time in the cell. The reaction proceeds in a controlled manner due to the specific nature of the enzyme specificity. I will talk about specificity of enzyme as we go on in this class, but not this particular class, in another class. A single enzyme will catalyze only a single reaction. That is specificity. If enzymes serve to control the chemical reaction that occur within the cell, and they ensure that they proceed to an efficient rate. The last thing I will talk about today is properties of enzymes. When you see enzyme, what are the properties? Enzymes are globular proteins. So enzymes are protein, but not all proteins are enzymes. So enzymes are globular proteins. Then, being protein, they are coded for by deoxyribonucleic acid. They are also a catalyst. Catalysts that involve the breaking down of substances. Another properties of enzyme is that their presence does not alter the nature or properties of the end product, in the sense that if it acts on a particular product and that product is oxidized to give another product, you see the enzyme will still be on its own. It cannot interfere in the product that you are going to get. Another properties of the enzyme is that they are very efficient. Very small amount of catalyst bring about the change of a large amount of substrates. Let's take for example, if one molecule of the enzyme catalase can catalyze the decomposition of about 600,000 molecules per second of hydrogen peroxide to water and oxygen at body temperature. Another one I want to talk about this enzyme is that they are highly specific. An enzyme will generally catalyze only a single reaction. Catalase example will catalyze the decomposition of hydrogen peroxide. The catalyzed reaction is reversible. Like I told you, it can be reversed, especially when you leave it still in that state. Their activity is affected by pH, temperature, substrate concentration, and enzyme concentration. Another property you need to know about enzyme is that they lower the activation energy of the reaction they catalyze. And the last one I want to mention in today's class is that enzymes possess active sites where the reaction takes place. And these sites, they have their specific shape where they bind for the reaction to occur. Thank you for today's class.